Welcome to this lecture on bacterial cell wall. In this particular lecture, we shall be discussing the cell wall structure of different associated bacteria-like organisms. We will start with the acid-fast bacteria, then we will be discussing the L-form bacteria, then we will be discussing the cell wall pattern of actinomycetes, chlamydia, rickettsia and mycoplasma also. We will be mainly discussing their components, their function and how actually these functional component impart pathogenicity to the pathogenic bacteria-like organisms. We will open our discussion with the structural pattern of acid-fast bacteria, and this bacteria is actually the causal organism for tuberculosis, and you know this organism is called Mycobacterium tuberculosis. This bacteria is actually having a multi-layered wall. You can see in this slide the external layer is formed of free mycolic acid and polypeptide, and it is followed by a layer of mycolic acid only, which is followed by the arabinogalactan, and then there will be the peptidoglycan. That means this peptidoglycan definitely will impart the gram characteristics to the bacteria. At the same time, it is having tetrapeptide, so the bacteria can also retain gram stain. But because of this external wall, it can retain the stain even if it is washed with acid alcohol mixture. So thereby this type of cell wall is called acid-fast cell wall, and the organism is called acid-fast bacteria. And this particular peptidoglycan is followed by the cytoplasmic membrane with an intermediate space or the periplasmic space. Now these particular layers, there are discrete 1, 2, 3, 4, 5 layers. They are interconnected. The most important backbone is the lipoarabinomannan or the LAM, which is connecting the outermost and the innermost layer of this particular bacterial cell wall. At the same time, there is also phosphoinositol manocytes, which is connecting the peptidoglycan with the cytoplasmic membrane traversing through the periplasmic space. It is more clear in this slide where we can see that this is the cell wall structure. You can see it is really thick, and this is thicker than a normal peptidoglycan bacterial cell wall. There will be large number of projecting molecules, projecting like flower plants in a garden, and these plants will include the mycolic acid, the glycolipids, also the mannose cap and the phosphates and the polypeptides. And you can also see that the size of these molecules also vary. Some are bigger, some are smaller, which definitely indicate the dynamism in the cell wall. And most importantly, because of these structures and because of these porine structures, they are well connected, and at the same time they are pathogenic in nature. The most important function is it is protecting the cell wall from osmotic stress. At the same time, it is also imparting pathogenicity. Because of the less porine, it is definitely not allowing any antibiotic to enter. So this explains how this bacteria cannot be acted upon by normal antibiotic. We cannot treat tuberculosis with normal antibiotic. And at the same time, it can also have outer molecules of adenosine, which will help in the attachment to the bacteria or to other host cell surface. And most importantly, it is actually inducing some inflammatory response. It is highly pathogenic. It is inducing some specific pathogen associated molecular pattern in the host cell. It is recognized by specific receptors, and after it is recognized by these receptors, the TLR1 to 6, they are inducing some unique factors generated within the cell, and these factors include the necrotic factor. So definitely tuberculosis will be marked by different tissue damage, and these are actually explained by this pathogenic marking system present in the bacterial cell wall. So in one way we can say this particular bacterial cell wall is imparting pathogenicity to the bacteria, and at the same time it is helping the bacteria to remain for a longer period within the lung, and as it stays in the lung, it makes the outer membrane in the form of tubercle, and thereby the disease is called tuberculosis. And actually the bacteria is being protected by the lung tissue, and at the same time the bacteria is protected by this unique thick dynamic cell wall. So the bacteria becomes virulent and it can cause infection and damage the tissue for a long time. Next important bacteria is the L-form bacteria, where the cell wall is absent. As you can see in this particular slide, that the cell wall is not there and the bacteria looks like a naked protoplast, and it is actually dividing also freely when grown in culture.
Sir, I have a question. Why do L-form bacteria lack cell wall?
You see, it is not a natural structure. It was denoted by Noble in Leicester Institute in UK and actually used the cell wall biosynthetic inhibiting antibiotic or enzymes like lysozyme, and is stripped of the cell wall. So when the cell wall is stripped off, it was producing spheroplast or protoplast, and they were dividing freely. And it can create stable structure, meaning they will remain like that for a longer period, or at the same time they can be unstable, that is they revert back to their normal bacterial cell. Now this particular L-form was created with a definite intention to see the pathogenicity of different bacteria. You can actually create L-form in case of E. coli, which is gram negative. It can be created for Bacillus, it can be created for other pathogenic bacteria as well. So even you can create L-form for tuberculosis bacteria, that is the acid-fast bacterial cell wall. So we can understand in a better way how this bacteria is invading and causing pathogenicity in the host cell. At the same time, this particular protoplast will join with each other. So this will give an idea of developing bacterial association and communication through quorum sensing. And at the same time, this particular bacterial cell wall is absent. So the receptors is absent, so they can evade the normal pathogenic or immunogenic reaction in human body.
Sir, I have a question. Sir, this L-form bacteria do not have any cell wall receptor.
Right.
So how does the body provide immunity against them?
You see, the body can actually protect from a bacteria by some specific phagocytes. So it will phagocyticize the particular bacterial cell, and at the same time this particular phagocytes cannot detect the bacteria because the cell wall receptor is lacking. But at the same time the membrane is there, the membrane protein is there. So against that particular protein or antigen there will be antibody produced. So in this way the lymphocytes will come into the saving this particular body from infection. This archibacteria, as you can see in this particular slide, it is a monoderm, that is a continuous layer of cell wall outside the cell membrane. And at the same time you can see also the gram negative and gram positive bacteria, where there is definitely the positive is monoderm and the negative is diderm. But the diderm meaning that there are outer and inner cell wall, where there is no outer and inner cell wall in archibacteria. Though at the same time, considering the different structural component, we can actually say that the archibacteria is much more close to gram negative rather than gram positive, because they do not have true murine, they have pseudomurine structure, because they are lacking the N-acetyl muramic acid and also they lack the D amino acid in the tetrapeptide. And this particular bacteria, you know, is definitely much more close to this gram negative bacteria. You can see in this slide that in gram negative bacteria there is this particular S layer, outer membrane, peptidoglycan and cytoplasmic membrane. Similarly, there is also the cytoplasmic membrane and the S layer, but truly there is no intermediate peptidoglycan. Whereas in case of gram positive, you can see a well defined peptidoglycan layer. So this particular cell wall is definitely having well defined S layer or surface layer. It is formed of specific protein molecules, and this protein molecule may be about 40 to 200 kD in their molecular weight, and they are actually creating some unique molecular pattern. And this structural symmetry, when we see, we really are astonished, because they will show different symmetry, like triangular symmetry as seen in case of P3, P4 is rectangular, and some thermoproteus, which is a thermotolerant bacteria, it can go up to hexagonal symmetry. So this type of symmetry is a unique structure of the S layer of the pseudomurine of archibacteria. And in addition to normal cell wall component, there is a particular component called methanochondroitin, which is present in some methanogenic or methanosarcina bacteria. And this actually help them to survive in a better way in cold or acidic environment. And this methanochondroitin is actually a trisaccharide formed of galactosamine and glucouronic acid. It is really very interesting to note that this particular monolayer or monoderm of archibacteria is protecting the bacteria from different natural stress in terms of acidity, in terms of alkalinity, in terms of high temperature stress. They are definitely helping the bacteria to survive in stringent environmental condition, and that is the reason why archibacteria actually could survive at the outset of life in this earth. And at the same time, this is having some pseudomurine structure. So we say normal bacterial cell wall may be the derivative of the old pseudomurine structure, though there is huge difference between eubacteria and archibacteria. And most importantly, as I was telling you, that there is definite protein molecules present in the S layer which is acting like a molecular sieve. The rickettsial cell wall is also having the peptidoglycan with the diaminopimelic acid, and most importantly, unlike the gram-positive bacterial cell wall, it is not having the teichoic acid, or rather it is more close to the gram-negative bacterial cell wall. And you can see this particular bacteria will have three layers. There will be the outer layer, or which is similar to the S layer in gram-negative bacterial cell wall, and then there will be the electron dense rigid cell wall, of course without teichoic acid, and there will be the inner cytoplasmic membrane, and there may be different amino acids present. And at the same time, this amino acid, if we consider totality, then they will come to about 0.5 to 6% of the total cell wall only. And this rickettsia is pathogenic in nature. It was denoted by rickets. It is causing influenza or typhoid-like symptoms in human body. So it is highly pathogenic. This is the typical structural unit of the rickettsial cell wall. It is having the normal NAG-NAM connected by glycoside linkage, having the L-alanine, the glutamate, the mesodiaminopimelic acid and D-alanine. But there is no teichoic acid, and this particular organism is pathogenic. It will enter the cell and will cause definite symptoms.
Sir, I have a question. How rickettsia escapes phagocytosis?
This is a very good question. Actually, I was about to say this particular point. You see, rickettsia will enter normally through the endothelial cell. Phagocytosis has to be induced. Now, when this particular rickettsia cell is inside the phagosome, then what happens is the particular rickettsia develop some typical actin tail, which is a protein, you know, a muscle protein is actin, and this is actually acting as a motor protein which will help the particular bacteria to evade away from that particular phagocyte. So what actually is done, before the phagocyte can release the enzymes to kill the rickettsia evade from the phagocytes, and thereby they are free in the blood and will cause infection. The chlamydia used to be known as viral particle earlier because it is lacking proper cell. It is only having an outer coat and inner cytosol. So it was actually known as non-infectious elementary body. But later on it was found out that this elementary body can enter into the host cell and cause infection, and they were called the reticulate bodies. So this chlamydia as such is having a cell wall similar to gram-negative bacteria, and they are actually having this typical two-layer system. That is, there is an outer layer and an inner layer with intermediate periplasmic space, and this particular rigid cell wall is definitely protecting the particular chlamydia when they are present outside the host cell. But when they are entering the host cell, they are actually facilitated by this unique endocytic process. You can see in this slide, this is how a chlamydia will attach to the particular host cell wall or host cell membrane, and they will actually follow a specific receptor, and then they gradually enter into the host cell, and then finally this particular elementary body will be transformed to the reticulate body, which is causing infection, and they will undergo division, and they are typically vesiculated, so they are not easily acted upon by the normal system of phagocytosis or other mechanism of defense, and then this particular cell or vesicle will enlarge inside the host cell, and finally they will come out of the host cell and will be coming out into the outer environment, either inside the body or outside the body. And that means this is how they percolate their life cycle. between elementary and reticulate body. That there is a typical gram-negative cell wall. You can see the outer membrane is having the LPS layer like gram-negative. Then there will be the protein channel or the porine channel, followed by the CRP layer, a small pre-layer or sort of peptidoglycan layer, followed by the inner membrane. And this inner membrane is actually associated with the cytosol and is also having unique integral protein for transport. The chlamydia particle or the chlamydia cell is having some unique projection on their cell wall. One such projection is the hemagglutinin, which is a protein having a definite head, a long stem with a cleavage site. Now by seeing this hemagglutinin structure, can you tell me which other organism might have this hemagglutinin?
Yes sir, it is found in influenza virus.
Yeah, that's perfect. You see, hemagglutinin is present in influenza virus, which really shows that chlamydia is very close to virus and it is having two important cycle in their life. This actinomycetes is intermediate between bacteria and fungus. You can see it is a filamentous structure with definite sporulating structure, and this actinomycetes is having a typical wall having muramic acid, and there are two definite amino acid, the LL diaminopimelic acid and the mesodiaminopimelic acid. Not only that, you can see this projection. This projection is actually the pili present in actinomycetes. And this pili can be of two types, type 1 and type 2. Type 1 is actually helping the actinomycete to bind with the host cell surface. Like in the mouth cavity, the actinomycetes may bind. At the same time, the type 2 is mainly helping in the aggregation of the individual actinomycete cell. And depending upon the bacterial cell wall or the cell wall pattern of actinomycetes, it was Cummins and Harris in 1958 denoted 5 different types of actinomycetes. They are the actinomyces type, the nocardia type, the actinoplen type, the streptosporan type and the streptomyces type. Now, how actually they have divided this? It is mainly on the basis of the presence or absence of certain diaminopimelic acid or other amino acid, or the presence or absence of some sugar residue like the pentose sugar. It is present in some form, absent in others. You can see this is the filamentous actinomycete. It is commonly called the Streptomyces cellicolor, and there is a triangular structure. This triangular structure is the SSGB protein, which is from a particular actinobacteria or actinomycete called Thermobifida fusca. This particular protein is from a different actinomycete and this is a different actinomycete. But what happens is this protein purified from a different actinobacteria, when is grown along with this actinomycete, it will impart sporulation and division in this particular organism. It indicates that they have actually unique aggregation where one protein of one organism can be functional in another organism and can help it to survive. This particular protein is similar to higher organism mitochondrial or eukaryotic mitochondrial protein. So what we can conclude that this particular actinomycete genome might have some role in the origin of mitochondria in higher eukaryotic cell. There is definite cell wall, definitely there will be receptor, so they will take up signals from the environment, and at the same time the cell wall will protect them. It is actually growing like a colony in a very stringent environmental condition, and it is also having some intermolecular action or signal reception growing in an aggregate or growing in a colony. This mycoplasma is actually the smallest individual organism, but this mycoplasma is not having a definite cell wall. This mycoplasma is having a membrane sort of thing, where the membrane is multi-layered, the lipoprotein membrane, which is formed of 3 layers, but there is no definite cell wall. And this lipoprotein membrane is having some unique protein molecules. These protein molecules may help in their locomotion or may help in their adhesion. Accordingly, they are called the Neymar protein or the adhesive protein. And their size is approximately 200 to 300 nanometer long. And this particular protein structure actually help them to survive as an animal cell, so they can change their shape, and accordingly you know perhaps that these mycoplasma are also called pleuropneumonia-like organism because they cause pneumonia on one hand and at the same time they are pleomorphic. They can change their shape because there is no rigid cell wall. Now, what these particular proteins are having specific function, and these functions include locomotion. This particular figure where the protein function of GL349 is denoted. This particular protein is binding with the sialic acid residue of the particular cell surface or receptor surface, and this particular protein becomes straight, and this is definitely ATP dependent, and it will actually bring about a conformational change in this particular protein which is creating a stroke. Like when we walk, we are striking the ground surface. So this protein is also striking the ground surface. In the next stage, the stroke is increasing, that is the leg is increasing in length. So the power of the stroke increases, the force increases. At this point the organism can move, and then what happens? We are moving, so organism also moves. How? This will be retracting. That means the sialic acid residue with which it has bounded, it has been released. So this organism retracts this particular tail protein and they go back, and again this cycle is repeated. So this is how this particular organism or mycoplasma is showing definite movement, and by seeing this movement we can actually compare this movement with an organism. Can you tell me which organism? It is the amoeba. So it is a sort of amoeboid movement where the cytosol projection is helping in their movement. So they are exhibiting amoeboid like movement. So the cell wall definitely is imparting this type of amoeboid movement, and at the same time they are helping in identifying individual receptor on the substrate or the receptor on the cell surface where it will attack. So in conclusion, what we have learned today, we have learned the different cell wall structure starting from the acid-fast bacteria, then the L-form bacteria, then the actinomycetes, chlamydia, rickettsia, and finally we concluded with mycoplasma. In this lecture, I have not considered cyanobacteria because cyanobacteria to many people, it is belonging to an algae. It is considered as a prokaryotic algae or cyanophyceae. Is it clear? Good.